Good evening, ladies and gentlemen, and welcome to the first of 3 webinars from the British Bee Veterinary Association. My name is John Hill. I'm president of the British Bee Veterinary Association, and also my sins, I'm also involved with Ulster Beekeepers, who are in fact helping me this evening to stage these webinars. Our lecture this evening is from Professor Dahliel Freitag from Austria, and it is today to do with the vaccination of a beehive. Now, what at this stage I would like to say is that our sponsor this evening is Vita Europe. Vita are in fact specialize in bee medicines, and indeed we would just like to show a short video of their company just now. Right, and again, as I say, thank you very much indeed to Vita for helping us to stage this particular webinar. Now, if I could introduce you to this evening's speaker, and it's really Dahliel Freitag is the associate professor at the Institute of Biology at the Karl Franz University of Graz in Austria. She has been studying insect immunity for over 15 years, I think initially at Helsinki University, and her desire to understand how organisms adapt to stressors has led to the paradigm-shifting discovery of the underlying mechanisms behind immune priming in honeybees. She is currently leading the honeybee pathology lab at the university and is teaching students about immunology and social insects. Outside of basic research, Dalial is a founding member and CSO of Dalial Animal Health Incorporated, a company pioneering the development of the first vaccine for honeybees. So, ladies and gentlemen, it gives me great pleasure to introduce Dahliel Freitag.
Thank you so much for this kind introduction. And yes, let me just share my screen. What suddenly my presentation disappeared. Here it is. And can you see my screen? All good?
Yes, indeed. That's grand.
Fantastic. Like I said, I've been studying insect immunity now 15 plus years as a woman. Let's not talk about the age. When I started to study insects many years ago, I remember I visited my grandma, and she was like looking at me in a very worried eyes and said, "Oh, so you're going to study biology and you're going to work with insects?" And she was like, "But don't you want to do something like meaningful in your life, something useful for society and people? And why don't you study medicine and become a real doctor?" And I was like, "No, I want to study biology and I want to work with insects." And now today I have to say that though my grandma has passed away many years ago, but I could tell her that actually I did become a doctor after all. It's just I became a doctor for insects, not for humans. So do we need doctors for insects actually? There are times when I started to study insect immunology and work with insect diseases, but insects were mostly regarded from a point of view, and insect research, especially immunology, was targeted towards getting rid of insects. So it's like the first question, so you want to kill them, right? And that was never my aim. My aim was really to understand how they are coping with stressors, how to make them more resistant against infections. And now, indeed, 15 years later, I think we really need people who understand insect immunity and insect physiology and how insects are coping with diseases. Why do we need it? Because, well, insects are disappearing. And while there are different reports on that, and a lot of scientists are still arguing how many insects we have lost and are all the measuring methods correct or not, we are estimating that we have lost 70 to 80% of insect biodiversity and biomass. I guess a lot of species have not even been described. And yes, also I'm sometimes secretly happy for having less mosquitoes, you know, when the evenings when you're trying to barbecue. And it was mentioned I spent a long time in Finland, and yeah, it's a land of mosquitoes, and summer evenings with barbecue after sauna are really hard. Well, last year's it's been rather pleasant. There are not so many mosquitoes around there anymore. Yeah, so, and while we are secretly happy without some of the more nuisance causing insects, we are also losing, of course, insects that are extremely important for our way of life, for our quality of life. And next to mosquitoes and horseflies, we are also losing pollinators. And not only wild pollinators, but also our number one pollinator on a planet, and that's honeybees. And yes, humans will not die out of hunger. There will be still rice, there will be still grain, but what we will be losing are the high nutrient value crops, like vegetables and fruits. So basically, loss of pollinators still means malnutrition. Malnutrition doesn't mean that we starve to death, but we will be sicker and less happy for sure. So loss of honeybees and pollination services means malnutrition for millions of people around the world. Plus, a lot of the foods, what we take for granted, like apples and tomatoes, will become for a lot of people unaffordable, especially for the low income families. And this is not the situation where we want to be. And just to show it here once, because we're going to talk about honeybees here. But next to the honeybees, the other important species what are disappearing are, of course, the other pollinators, like different bumblebee species, but also the ants of social insects next to honeybees, and who are exposed to exactly the same type of stressors and reasons for decline as honeybees. And they are also extremely important parts of our ecosystems. We are dealing with dead matter and erosion of the soil. So, for example, if our crops would grow better. And these are all part of a big group of social insects. So it is dire need to understand how these insects are fighting the stressors and how we can help them to become more resistant against the different environmental stress factors. So why are we losing insects? So what is really happening? And I always say there are 3 reasons for it. Reason number 1 is habitat loss and nutritional deficiency. So what our pollinators, but also other insects are seeing in their environment is, well, they don't have anywhere to live anymore. A lot of environment has turned into huge fields of monocultures. And yes, a cornfield or a wheat field is nice to look at, but for a pollinator, it's a desert. There is nothing to eat for them. The same way we would not enjoy the buffet, there is only rice and nothing else. Or even worse, there would be something completely inedible for us. So basically we would be starving. The same is happening to our pollinators. Number 2, pesticides. In order to maintain these huge fields of monocultures, of course, we need to spray them, because this is basically open call for any pest species, but also for a lot of diseases. So a lot of fields are sprayed with huge amounts of fungicides and herbicides to get rid of other plants what might want to grow there. And though a lot of pesticides are claimed to be safe for insects, they really are not. Glyphosate, I think one of the most controversial herbicides of late, we know it actually has severe effect on pollinators and also other social insects. And what happens if your life quality goes down, if you're starved, if you're poisoned, then of course your resistance mechanisms are not anymore very good, and then the diseases come. So literally, if we ask what is happening to the pollinators, what is happening to the other insects, there are 3 things are happening. Nutritional deficiency, coupled with habitat loss, pesticides and diseases. That's why we are losing insects. That's why we are losing pollinators. And while we cannot, of course, tackle them all simultaneously, we need to start from somewhere. So what can we do about it? Let's have a look how a bee life looks like. So as a scientist, we always try to look at the problem from different angles. So what are bees exposed to? And how they were maybe a long time ago evolved to live. And this is what you see here is maybe something of a dream or some kind of a natural form how beehive would look like. As you see, here is also free hanging. A lot of bees would also nest inside of the tree trunks, of course. And it's not very big. There are not too many bees together. Now, what we've done with it is, we have industrialized bee life. And while these kind of pictures are not yet very common in Europe, in US, this has been happening already for decades. So the bees are grown in huge population sizes next to each other. There are literally millions of bees are commingling in one location. And now what COVID has taught us all is that if a lot of people are together without any social isolation, diseases are spreading, right? We've been encountering all kinds of interesting measures for the past 2 years in different countries. What's more, so a lot of these beehives are uprooted and put on a semi-trucks and driven to another county for thousands of kilometers where they meet with the other bees. So it's like kind of like a airport all over, again, all the time. And of course, you can imagine if there is a sick beehive somewhere, then this disease will spread among these potentially malnourished bees relatively fast. And we can have a huge epidemics spreading through the population. So mass rearing of animals, and this is exactly what we are doing with honeybees right now. It's a mass rearing, it's a real animal husbandry. But a lot of people do not see it this way yet. So I think our task is also as beekeepers and the scientists to make people aware that this is real animal husbandry. It should be treated like that. It's not people who just have one hive in their backyard. No, this is real industry. It needs more support and more understanding and more help on a higher level, because the mass rearing of animals has been happening for centuries, as you know, and there are very tight regulations, there are very tight norms to deal with some of the consequences, what happen if something goes wrong in these facilities. One of the things what can go wrong is that the diseases can spread among these mass-reared animals. And as you know, also in the chicken farms or in the cow farms or also in human populations, we need to deal with the consequences. But to help out these animals, what are being kept as a cattle, or also our populations, there has been this breakthrough technology. I still like to call it breakthrough technology, even if it's over 200 years old. What was discovered by or modernized by Edward Jenner, and it's called vaccination. What is vaccination? Vaccination is a controlled exposure to the selected antigens to induce specific immunity against a specific disease. And this is something what we use standardly all over the world. And we have oral vaccinations and we have injection vaccinations. And it's a rule and regulation in many places. Try to travel with your cat or a dog across the border. If you don't have a valid vaccination passport, trust me, you're not going to get very far. Moreover, we know that vaccination, the natural one, it's based on immunological memory. So basically, once we encounter the antigen, we develop a long-lasting memory against this disease-causing agent. And we also know that this can be transferred from one generation to another. We know that, for example, mothers are passing on these antibodies to their babies, but it also happens in birds or in reptiles and frogs. So this kind of immunological memory is transgenerational, and this transfer of antibodies, and is very useful in fighting against the different diseases. Now, what is the hallmark for this type of immunity is the presence of antibodies, like I said. And you see here on the right side, and there are fishes and pigs and snakes and children. And it takes time to form, of course, first. Now, if we talk about the honeybees, when you can see in the left corner, and honeybees and all the invertebrates do not have this adaptive immunity. They only have innate immunity. What means that they only have, or this is what we have been believing so far, they only have this type of immunity with what they had been born. And these are epithelial barriers, so basically their cuticle, but also different cells that are circulating in the hemolymph, or different proteins and peptides. But they do not have classical adaptive immunity, what is based on antibodies and for example, T cells. So this part is not typical for honeybees. That's why we also believe it's not possible to vaccinate them. Despite the fact that they do not have this sophisticated part of immunity, they still have very efficient methods to fight against the diseases, and I'm sure you've been hearing about it before. So how do bees fight diseases? They can fight it on individual level, and they can fight it at the colony levels. On an individual level, we have different physiological defenses like immune system itself. So like I said, synthesis of antimicrobial peptides, for example, RNAi mechanisms to fight off viral diseases. Then synthesis of phenol oxidase, that's an enzyme for example, helps to fight against fungal diseases, but also
Right. I think, Dahliel, unfortunately, your screen is frozen. Ladies and gentlemen, I think we'll just have to wait.
Of food.
Right.
But also they are able to eliminate sick individuals from the hives. But like I said, among all these sophisticated immune responses, what they do not have are antibodies. Nevertheless, so who is actually threatening honeybees? There are different fungal infections. One of probably important one here is, for example, chalkroot, what is spreading more and more, especially in Northern Europe. Different bacteria, Bacillus larvae, what is zero tolerance disease in the European Union in most of the cases, and different viruses. I guess I don't need to tell you about the deformed wing virus, what is thought to be one of the #1 reasons behind the bee declines. So our poor, starved, poisoned honeybee needs to fight off all these diseases at the same time, with having only innate defenses. So no wonder that it has difficulties to cope with all of that. So what do we do today if a bees get diseased, if they get infected? But insects in general, so it doesn't only apply for honeybees, but let's talk about bees today. So literally there are two ways, right? In some countries, you would soak the hives in antibiotics. And I guess we can agree it's not the best method in the world, because there are a lot of negative side effects of that. And among other things, the disease-causing agents are getting resistant against it. So the effects will not last very long. So the only thing what is left over is literally burning your hive. What I think for anybody who has bees or animals is not a favorable way out, because not only you're going to lose your animals, you're burning also your equipment or anything that has been in contact with this disease hive. So that is definitely not sustainable solution in the long run. So we need to find some alternatives. So how big are honeybee losses? So it's estimated that up to 45% of hive losses are due to different diseases. So we are talking about remarkable amount of beehives that are lost to different infections. And it's estimated that up to 1/3 of a profit margin for beekeepers is lost because of the disease in honeybees. But it, of course, only does not stop with beekeepers. As the next, of course, it affects the people who are growing different crops. So basically, no pollination means less yield for growers. And in the end of the day, it touches all of us, because less pollination and less crop means also less food, and decreased food security. I guess we can agree here all that this is touching every single person on Earth. And we're not talking here about production, just honey. We are really talking about foods that are essential for survival for everybody. So we are talking about the huge, huge problem. So what we believe, what beekeepers really need are better solutions. What the solutions should be, they should be scientifically proven. So we're not selling a snake oil. It needs to be proven. It needs to be real, going through the vigorous testing in a lab. So it needs to be a real solution. It needs to be safe for bees and beekeepers to be used. It needs to be effective in solving the problem. Preferably shouldn't be any harsh chemical solution. It should not be based on genetically modified organisms, and it should be compatible with organic beekeeping. So this type of solutions we are needing these days in beekeeping industry. So here, of course, I think this ball over to us scientists. So what can we do about it? Let's think, let's try to solve this problem, because when I'm asked, so why do we need scientists in this world? One reason for it is we are the problem solvers, and bees are dying, they have a lot of diseases. What can we do about it? So one thing what we can do is we can try to understand even better how the immune system works. So I told you before, it is very complex on different levels. What else do we know? So let's look at let's zoom in on an individual level. So if we talk about bacterial diseases, how does the immune system recognize and react to a specific bacterium, what, for example, honeybee queen is encountering? Like any bacterium, for example, American foul brood is ingested with a food. So basically, the organ what is encountering the bacteria first is digestive system. What happens with bacteria in the gut is actually they are broken down into pieces. So now these bacteria encountered here are then taken into pieces. The pieces are taken then later to the humuscle, so they are in the body cavity. There, the species of bacteria are activating specific immune receptors in the fat body cell of a honeybee queen. There they know that they are triggering synthesis of specific antibacterial peptides, but also synthesis of a certain immunological cells. So this is what is triggered when the queen or also the worker is encountering the bacteria in their diet. What else is happening in the fat body is that fat body is the place where vitellogenin is synthesized. Vitellogenin is the oldest egg yolk protein on Earth, on animals. And it's present in all the egg-laying animals, so also among others in a chicken, so that's why there's a chicken egg. So if you think about vitellogenin, so the next time you're gonna have your egg in the morning or in the evening, how you like it, then the yellow of it is vitellogenin. What is vitellogenin? It's important actually the development of a larvae and the embryo in the egg. So vitellogenin is packed into the developing eggs, and it forms nutrition for a little bee inside of the egg. And if we talk about the dry weight of the egg, then about 92% of the honeybee egg dry weight is vitellogenin. Like I said, important fact is that this is synthesized in a fat body. And fat body, fat body is, I wanted to go back here. Fat body is exactly this place where also the species of bacteria are taken up in the case of the honeybees. Like I said, present in all the egg-laying animals on Earth. What we also know is that honeybee vitellogenin is able to bind to pieces of bacteria. So basically, what are the digestion products of encountered bacteria? And if you look here, so we carried out very fancy assay. It's back in the day when I was working in Helsinki with a colleague of mine. This PG is actually characteristic for a cell wall of gram-positive bacteria, such as American foul brood. So as you can see, vitellogenin binds very efficiently to the pieces of American foul brood. Further on, we carried out also in vitro assay where we showed that vitellogenin is important not only binding to these bacterial pieces, but we also showed that vitellogenin is able to take these pieces to the developing eggs. So if we zoom into, here is a histological cut of the developing egg in honeybees, then you can see if vitellogenin is present, then we see uptake a lot of labeled bacteria in the eggs. So literally what vitellogenin is doing, it's kind of like a transport mechanism for the, if you like, natural vaccine from one generation to another. So it's a mechanism behind transgenerational immune priming, what is not based on antibodies. So we don't need these fancy antibodies here, but actually vitellogenin is doing this function. It's carrying, if you like, then we could say it like this, that the pieces of vaccine from one generation to another. And now when the larvae is, of course, being born, it's already encountering these specific bacteria that mother has been exposed to. So this is kind of natural vaccination. And now when the larvae is born out of those eggs, then of course it has already all these mechanisms what will help this larvae to fight against the diseases upregulated. So with the studies done in vitro, so in a lab, we dissected the ovaries out, we put them in different solutions, and we showed that in a fancy imaging way. But you would tell me that, hey, but you know, how would it work under natural conditions? Because the queen doesn't really feed herself much. That is correct. Because queen is fed by the worker bees, right? So in order this vaccine really, or this transgenerational immune signal reach from queen to the larvae, it first needs to go through the worker bees, right? So for this first, this signal and these little red dots here need to go through the worker gut as well. They need to be taken up by the fat body in a worker, what is also placed for vitellogenin synthesis. Only in workers, it's synthesized in order to be transported to the head glands or hypopharyngeal glands, where it's incorporated into the royal jelly, what is fed to the queens. So in order for this immunological signal to reach the queen, it needs to go through the worker. And we set up the experiment to see if that is actually happening, if that's possible. So first hurdle would be going through the midgut. And as you can see here, you see these little pink dots. And we've shown that, yes, the labeled bacteria and pieces of bacteria can go through the worker midgut. Next, can we see them in a fat body? And indeed, you can see here is a nice, beautiful fat body cell. And you can see that these labeled pieces of bacteria are taken up by the worker fat body. And now, of course, very important. Can it reach the hypopharyngeal glands? And yes, it can. Again, here is an image of a cell in the hypopharyngeal glands, and you can see this nice pink dots, what depicting the labeled bacteria also present in the head glands. So yes, it can go through the worker organism, and it can reach the target tissue in this case where it needs to go in order to be put into royal jelly and fed to the queen. And next, of course, final frontier. Can we see it also in the ovaries of the queen? And yes, indeed. So on this left side, you see the image of her ovaries from a queen who's been fed with labeled bacteria. In this case, this was actually labeled American foul brood. And you can see if the workers have been fed with it, we see the accumulation of this fluorescence and the signal also in the queen ovaries. We do not see this kind of bright fluorescence in the control case. So yes, indeed, the signal can go from the workers to the queen. So now we understand more and more about immunity in honeybees. And this should allow us to develop more resilient ways to fight diseases in honeybees. And how can we use this understanding to design the vaccine, the real vaccine for honeybee queens? So in this case, since I showed you this pathway, it's an oral pathway, right? So it's fed to the honeybee queen. And the way we see it, that the vaccine can be put in the liquid solution and can be mixed to the queen candy, what is already present in all the queen cages, right? So that if you're shipping a queen from one location to another, you have usually this kind of sugary paste here. So the oral vaccine can be added to this paste, and basically the queen is being vaccinated while she is housed here with the nurse bees. And then, of course, once the queen is entering the hive, she would be laying eggs that have already been primed immunologically. So does it really work? So how do we do our trials in a lab? Just showing you a little bit our kitchen side. So which kind of proof do we have, and how do we get this proof if it really works? So the experiments we are setting up are based on vaccinated queens and non-vaccinated queens. So we have always placebo-controlled experiments. So we have one set of hives that's going to get vaccinated queen, and the other set of hives will get a control queen. So after some time in a queen cage, when the queens are transferred into a normal hive, where they are allowed to establish the colony and do whatever queens do in the hives. And after a while, we go out and check if we have already eggs and larvae present. So once we know that there are larvae present, we bring really about 24 hours old larvae in the lab, and we craft them in the Petri dishes where we are feeding them with artificial diet. And so we place the little larvae in these droplets, and then we expose them to the control treatments, and then we challenge them with virulent bacteria to see if this kind of clean vaccination has really enhanced their resistance against the disease. We, of course, cannot do it in a field because we are working with American foul brood in this case, and I cannot go to the hive and spray it with spores. I would be probably fired and put into jail if I do that. That's why all the experiments we are doing, we're carrying it out under laboratory conditions. So what we would like to achieve with this kind of immunological priming is that we would be able to protect against specific diseases that wouldn't it be nice if it would also protect against other diseases, to have certain cross protection mechanism, but also increase overall performance of the hives. So let's have a look. Can we achieve protection against specific diseases with this type of transgenerational immune priming mechanism? And here, so we have two sets of hives. So we have here unprimed hives, and then we have primed hives, and we used oral formulation with American foul brood. So we use here heat-killed American foul brood bacterium, what has been embedded in a queen candy. And what we clearly see here, that the larval survival is way higher if they come from a hives what have been established by the primed queen or by vaccinated queens. So if queens are not vaccinated, then mortality is way higher in this case. So we see the clear benefit of immunological priming via honeybee queen. And we don't really see any negative effects of the priming at the same time. So we set up a similar experiment to see if it would also work against European foul brood. So again, we had a set of hives where we didn't have any priming, and then we had a set of hives where the hives are primed against European foul brood. So it was similar oral formulation with a heat-killed bacterium. And results here as well are very nice. You see that in the case of vaccinated or primed hives, the larval mortality is literally just the background mortality. So one would say that the priming here gives literally absolute immunity against this disease, what is very, very promising. So next, I said, wouldn't it be nice if priming against one disease would also give to some level cross protection against other diseases? And indeed, so we checked in this case a cross-protecting effect against American foul brood if we primed the hive against European foul brood. And here on this right corner, you see the larval mortality in the case of a priming against European foul brood and the infection with European foul brood. Now, when we infect the larvae coming from same hives, yes. If we infect this larvae with American foul brood challenge in the laboratory, what we see that maybe effect is not as strong as with European foul brood, but we still see considerable improvement in survival also against American foul brood infection. So we have a real cross protection effect here. Next, I said, wouldn't it be nice if we would also have certain increase in overall performance? So, and here, this is a study done back in Finland a few years ago, where we had set of hives that started at the same level. So we had the primed hives and control hives, and here the priming was against American foul brood. And over the season, we see about 2-frame improvement in the primed hives. So we have 2 full frames more of bees before we went into overwintering. And though this result is not statistically significant here, we see a clear trend that the hives would have been primed, maybe slightly performing better. So I mentioned before that safety is important in this kind of experiments. And what is the queen survival of such treatment? And I think over the years, I've been probably doing this priming treatment or vaccination treatment, I think about 500 queens for sure. And I literally do not see any negative effects. So it doesn't matter which kind of priming treatment we are doing with the queens, the survival is almost always 100%. But you lose random queen here and there is only normal. The other thing we also checked was the honey quality. And these are some standard things what I checked in Finland before you want to sell honey as a commercial beekeeper. And so far, again, we have not seen any negative effect on honey quality or honey production in vaccinated hives. So we believe that using this mechanism, what we understand now, and what we call transgenerational immune priming, we can create new strategies to manage honeybee health. And this type of controlled priming can protect against specific diseases, like I showed, increase also defenses against other diseases, like I showed American foul brood, European foul brood, but also it has potential to increase overall performance in honeybees. And so, like mentioned before, I am a founder and a chief scientific officer in Dalial Animal Health. So Dalial Animal Health is utilizing these research results that I presented today and is in a way of developing the very first vaccine for honeybees. And we're targeting here American foul brood infection. The vaccines that Dalial is developing will be regulated by the regulatory organizations. So that is, for example, Center for Veterinary Biologics in US, and it will be in Europe. And if it gets approved one day, it will be regulated product, it will be vigorously tested product, and it will be very first vaccine out there for honeybees and for insects in general. And with that, I would like to thank you, and I will be happy to answer any questions you might have.
Dalial, thank you very, very much indeed. That was absolutely fascinating. And I think it is a remarkable development that we're actually able to move on to, say, to vaccines for invertebrates. And certainly, I think, as you say, you're involved with a company. I think you're involved with. It's called Dalial, which is seeking a license in the United States at the present time, with the help of a large veterinary pharmaceutical company, and see how that will actually, if hopefully you will get the license in the States, and then hopefully you will go to Europe. So. Certainly, I think this opens up, hopefully, a very large number of particular questions. So, ladies and gentlemen, if you would wish to put your questions to Dalial just into the Q&A box, and we'll take some time to see if we could answer them. Yeah, I think really the first one really comes up here, Dalial, is it true that honeybee numbers are not dwindling, but most other bees are?
This is, honestly, I don't have the latest data on the hover pollinators, but of course, hover pollinators are affected as well. Like I said, insect losses are worldwide reported, and among others, also wild pollinators. And I know honeybees are getting a lot of bad rap here, that they are like, causing the decline in wild pollinators. I do not believe that is that simple. But what we are seeing, that the bee numbers are actually declining. yes, there might be more and more beehives, but bee mortality is quite often higher than before. And yes, we have sometimes good years, but we also have some very, very bad years. So especially troublesome or worrisome for me, what I've been seeing lately are the problems with queens. I mean, last couple of years, we've had tremendous amount of queen losses for very random reasons. So the queen failure is increasing, I think. So while we are still managing to produce new queens, I've also talked to some beekeepers in US who believe it's not sustainable soon anymore because they just can't produce enough, as many as needed. So I think the pollinator losses in general, both wild bees and honeybees, are going hand in hand.
Yeah, yeah. I would think certainly say the problems also related to bumblebees and to solitary bees is that they are, a lot of those species are certainly struggling. I think that would
they are.
Yeah.
They have the same issues with honeybees. It's also the monocultures, it's also the pesticides and diseases. And yes, of course, there are also crossover diseases from honeybees to the wild pollinators. So if we would manage to create the situation where the honeybees are healthier, then we would also spread less diseases to the wild pollinators. I think this, this goes without saying, right?
Right. Okay. This is just a by the way sort of question. It really said from Robin, he said that I saw in the Vita video the mention of Fumagillin B being reintroduced for use in Canada. Is it likely to be available again in the UK? I don't know whether you're aware. Are you familiar with this particular one? Possibly I can answer that because I was involved. Say, the UK was probably the last country, in fact, to ban the use of Fumagillin in Europe. There were various reasons for that, and which was to do with efficacy, and also I think that there was a very, very slight suspicion of potentially carcinogenic properties. Now, as soon as any regulator, certainly in the UK, hears of anything that could be vaguely carcinogenic, there instantly would be banned, and that's the way it is. I'm not really quite sure what's happening in the United States. I'm in Canada for the reintroduction of Fumagillin. I'm afraid I have no knowledge of that at all, but I think it's probably most unlikely that it will be reintroduced into the UK or Europe. So sorry about that.
John, is it worth asking if we have anybody in the audience from Vita? Or I'm not sure if there is or not, but
I'm not sure.
It's a question for them, really, isn't it?
Is if they want to, if there is anybody in the audience from Vita, if they want to comment, maybe in the Q&A box, yeah, or the chat, yeah. Anyway, if we can move on to the next question, it's from Graham Kingham, who in fact has written some books on bees in recent times. And he said, given the large quantities of hives in close proximity of each other, the risk of disease spread is far greater than, say, in wild colonies. Are we going to be in an arms race with the diseases, just like Varroa treatment becoming tolerant?
Of course. I mean, that's the clear thing from a sense. If there's higher density, and as soon as the disease is entering the apiary, it has high potential to spread among all the beehives, because quite often, and I guess you know it probably even better than me if you've been beekeeper for a while, because like I said, I have my research hives, but what we see clearly is as soon as one hive is getting a bit weaker, whether it's because of disease or anything else, the other bees would go in there and start robbing the honey, for example. And what they bring with them is not only honey, but also the diseases, especially diseases like American foul brood or child brood or European foul brood, but especially anything sporulating. So because here all the adult bees are vectors, right? So they don't get sick, they don't get problem, but they are perfect vectors and carriers. So yes, we are arms race from this point of view, and I know that the child brood and European foul brood has not been quarantineable disease so far in Europe, but I've heard now that the new recommendation in many countries is just burn the hives also if it has European foul brood, because it's so hard to manage it. So we are in arms race, because the more stressed the beehives are, the less they have a chance to deal with it. And my own experience from the last couple of years also says, so the experimental setup where we had hives and pellets, like 4 hives in a block, if one got infected, then it really spread among all of them. It's just really hard to maintain. So you really need to isolate and deal with it immediately if you want to stop the spread.
Oh, yes, well, certainly I think that's very apparent, is that European foul brood is what we call a classical subclinical disease, and very often by the time you see the clinical signs of it, you have a lot of infection in the area. Certainly it is. Yes, we'll move on then.
I've heard this.
Sorry.
Yeah, I've heard the same for child brood lately. So yeah.
Yeah, right. Move on. Drones, do they pass on any immunity after the queen vaccination?
We haven't looked into drones, but I would expect no.
Right, okay. I think that's all right. Okay, now this from France. Thank you very much for a great presentation. In many countries, would this be allowed? Austria, Germany, or only outside Europe? What time horizon do you assume to get a license? I think that's a order.
That's a good question. So I will answer as diplomatically as I'm allowed to answer, because of course regulatory organs do not like if you say any timelines. We are, so first, our first market and target is US, because it's more consolidated market. And fingers crossed, we are in a very tight discussions with CVB, which just had actually a chat last week with them. I gave them a very similar presentation on our updates on clinical trials. So we really hope, fingers crossed, that the license will take months and not years. So we should be very, very close to be allowed to sell it in US. So our hope is that once it enters the US market, the Europe will come as well, because it's already used in US. We can show it's safe, and there is always emergency need pathway also in Europe possible. So we are hoping to enter European market rather sooner than later, because I know there is a need for it. So I cannot give a time horizon for Europe, but like I said, I hope sooner than later.
Very good, yeah. Thank you, fascinating talk. Does the vaccination need to be renewed as the queen uses up the virus, or well, be the virus or bacterial particles in egg laying?
What I can say so far, that we've been testing it throughout one season, and it definitely lasts one season. So you don't need to do it every month. Now, would you need to do it next year? I cannot say it today. The trials are ongoing on that one. But then again, what we've noticed being in chat with beekeepers, and actually maybe it would be lovely or potential to use your network to ask this. So how long do you keep actually queens? Because what we hear that a lot of beekeepers renew their queens every year. So what we can say that 1 year the vaccination will last.
Right.
So you don't need to do it in between. For 4 years, probably not. But who keeps queens for 4 years these days? I think very few individuals.
Does the transgenerational effect decline over time? I think that really is related to that last.
I guess it's linked to this, what I just said.
Yeah,
I would assume it to decline after maybe a year or 2 years, 3 years. But when we consider the lifespan of a queen, then I think we are playing quite safe here. I mean, the humans, we also need to revaccinate every 7-10 years, depending on a disease.
That's true, indeed. Could the vaccine protect bees from deformed wing virus?
That's a very good question, and it's definitely something what I'm planning to look into. We've been working so far with bacteria and fungi. I didn't show the data today, but we also have a proof of concept for child group, so it works exactly in the same way. And we can get very good resistance against the child group infection. Against deformed wing virus, what I know from the literature and from other insects, it should work. It really should work. It's just about having resources and showing and figuring it out how we can do it in honeybees. But I would think we can figure it out.
Right, okay. There appeared to be quite a lower response in the primed larvae to EFB compared to AFB. Has any idea why this might be so?
Okay, so what I showed you for EFB was LD30. So we only, in this case, we used like lethal dose of 30%, and in the AFB we used 50%. So that's why it looked so different, the mortalities in a way. So actually the response against EFB was a bit better from this point of view, but also you have to think about these are lab studies. We blast them with huge amount of spores, and we are trying to figure out what is a good LD to work with. So we would not reach LD 100 because that would just kill everybody. But yeah, we use low LD in EFB. It's very hard to work this bacteria with. It's really extremely hard.
Right, okay. This is from Janet. She said, could you just run through how the vaccine is prepared, please?
Okay. Yeah, so it's, I cannot say all the details, of course, but because that's a kind of part of the company, because right now anyway, we move to manufacturing. But in principle, we grow up the bacteria in the artificial medium in a lab. We collect them, we concentrate them, we heat kill them with autoclaving, so it's a pressure cooker literally. Then after that, we make sure that they are really dead. So we plate the bacteria out, we make sure that none of the spores have survived, and then we mix these dead bacterial fragments, because since they are heat killed with a pressure cooker, they just explode, and we mix it into a queen candy. And then we feed it to the honeybee queens.
Right, okay.
So it's a classic oral vaccination like we used to have against polio. I think as a kid, I got a sugar cube as a polio vaccine.
Right, okay. The next one is actually from Kirsty Stainton. She's a bee scientist in England. Have you tried using booster shots in your vaccination experiments?
Booster shots, I guess I need to ask, do you mean with injection of it or a double vaccination?
Well, I think whether it's maybe revaccinating, trying to revaccinate the queen. I think that's really what she's asking.
Okay, we haven't done it so far because we haven't had need for it, because like I said, we've been vaccinated. So basically we've been sampling from a hive until they went to overwintering, and we've still seen immunity against it. But in principle, of course, it would be possible. We could capture the queen and feed the candy again with the vaccine. So it's definitely would be possible, but we haven't tried it yet.
Right, okay. Next one from Michael. He said, a very basic question, I'm sure I have not understood, but I assume that vitellogenin is a non-specific antibacterial compound. So what do the specific bacterial fronts induce?
Okay. Yes, so I mean, you're absolutely right. Vitellogenin has actually like thousands of other functions in an organism. So it is not specifically targeted to bind bacterial fragments, but for some reason it does it, and it's a good thing, because that's why it can kind of carry naturally vaccine from one generation to another. So what really induces the immunity are these species of bacteria, what are result of a digestion in the honeybees. So like in our case, the bee digestive enzymes would kind of chop bacteria up into pieces, and they are taken up into the body and then carried to the eggs. And now when the vitellogenin is breaking apart in the developing embryo, because it's being used for food, these species of bacteria come free, and then they would literally initiate the immune response in the larvae. Like you would inject it into the egg, only you do not inject it into the egg. The mother kind of packs it as a dowry to the next generation.
Right. I think there's a number of questions to do with viruses. It really is, you know, can you move this particular technique onto virus? And I think you really answered that really by saying that
We're planning.
You're planning to do that eventually. Let's see if we come. There was. Whoops! I'm just past one there. I think. Yeah, really. It comes from Julian. My understanding is that Varroa is the cause of the greatest colony losses, either directly or indirectly through virus spread. How do you see bee vaccination helping with viruses that enter the bee, not via the gut, but via Varroa?
Well, I mean, in the end, we still need to prime the immunity, right? So if we find a mechanism how we're going to prime systemic immunity, because also in this case, right, so we do it via the gut, but the priming effect is systemic, right? It doesn't only affect the gut, it's just taken up through the gut in the queens. And the same would need to work for viral priming. And like I said, it's known that it works in Lepidopteran system. So the mothers who've been exposed to are laying eggs that are more resistant against virus. So we know in principle it needs to work. So I think it will still work also in the honeybees. And saying Varroa, I think Varroa and deforming virus is a little bit mosquito and malaria problem. So mosquitoes itself, of course, Varroa is also a nuisance, but if the bees would not get infected with the virus, they might also have a better chance to stand against Varroa.
Right. Okay. Let's see. We said Australia from how important is it that we all treat at the same time? I presume it means would you tend to have a season where you would treat queens all at the same time?
I think the beauty of it, you can treat them whenever you need to treat them, actually. Mm-hmm. So, of course, the most practical time would be when you are placing the new queens in the hives, so that the new cohort who would be created would be more resistant. But nothing speaks against it that you can do it also later. So we've done it at a different time of a year due to sometimes just practicality of experiments when they are happening, because I haven't talked about company research here, so it's all the academic research. And yeah, I've done it whenever I can, literally. I've done it in December in US, I've done it in July in Finland, I've done it in May in Austria. So it's really, it depends how you can. I've done it as late as I think also in the beginning of August in one of the countries, still works. Yeah, it really depends on a specific need of a beekeeper.
Okay, I think really, as to say, I think you've answered a huge number of questions. I think we will make this actually the last question. Okay, it's from Mark, and it said, it has been shown that Varroa are feeding on the fat bodies of the bees recently. Is it possible that your research could be useful to create a treatment for Varroa using a similar method to your virus priming? Interesting question.
Yeah, I actually know that paper. We just discussed it in my group a couple of weeks ago. I think that a super interesting paper, and I was also thinking that I should sit down and think about it. Maybe we could help with that way, because like I said, whatever we're feeding is taken up in a fat body, and this is exactly what Varroa is targeting. Wow. So that's good thoughts. Yes. It was a very interesting paper. I enjoyed it as well.
Well, Dalial, as I say, I think we'll read the last question, and I would like to thank you very, very much indeed for what was a superb lecture, and indeed to thank the audience for what was a superb number of questions. And indeed, and certainly I think this research and development will be carrying on, and I think we will be keeping great interest in it indeed, and we do hope that that you will have every success in your endeavors. So, ladies and gentlemen, thank you very much indeed for listening in this evening. I do hope that you found this a very interesting lecture, because really, effectively, it's the first time that has been broadcast in Europe, I think, and I hope that certainly that we will hear a great deal more about this in the future. And again, thanks to Vita Europe for their very kind sponsorship of this evening's lecture. And if I could just say is that this day week, next Wednesday, we will have Dr. Una Fitzpatrick from the All Ireland Pollinator Plan will be giving a lecture on that plan. Many of you may be aware, but the All Ireland Pollinator Plan has been generated by a small number of people and has been hugely successful in promoting the care and protection of pollinators right through the whole of the island of Ireland. And I think you'll find it will be a very, very interesting lecture. So you may look forward to that next Wednesday. Anyway, again, to thank Dalial again, and wish you all a good evening, and hopefully we'll see you next week. Good night.
Thank you so much.